Welcome to this video on protein translocation to rough endoplasmic reticulum or RER. Proteins destined for the plasma membrane, Golgi apparatus, secretion or lysosomes are typically translocated through the channel called translocon, which is present in the rough endoplasmic reticulum. These proteins are translocated co-translationally. It means the ribosome must bind to the channel. How this bind, what is the mechanism, what are the requirement, we are going to discuss that. But you should know that these protein must pass through this channel so that they can be modified extensively in the endoplasmic reticulum or rough endoplasmic reticulum, from where they join the Golgi apparatus for further modification, and finally they reach the trans-Golgi apparatus, from where they are sent to their final destinations such as lysosome, Golgi, secretion or plasma membrane. In this video I will review the requirement of translocations, including signal peptide, the signal recognition particle, the structure of translocon, and the mechanism of coupling of ribosome with the translocon. For understanding the protein translocation through rough it is important that we systematically and briefly understand the properties or characteristics of the components, including signal peptide, signal recognition particle or SRP, SRP receptor, and translocon. Let us discuss the features of signal peptide. It is present at the N-terminus of a protein, for example a secretory protein. On comparing the signal peptide sequences of different organisms including humans, a general structure is proposed. It has three regions: N region, which is positively charged containing positively charged amino acid, and H region, which has hydrophobic amino acid, and this region is believed to help in the penetration of peptide through hydrophobic membrane. This is rich in alpha helical structure. This region is followed by C region, which is basically the carboxy group, carboxy end of the signal peptide. This is followed by cleavage site. In many protein, it is often cleaved. The signal peptide is cleaved often, and therefore you will not see in mature protein signal peptide in many proteins. For translocation to a protein must have a signal peptide at its N-terminus. The SP or signal peptide basically connects a ribosome to the rough endoplasmic reticulum translocon through its interaction with SRP, which we are going to discuss in the next slide. Let us discuss signal recognition particle or SRP. It consists of 6 subunits which are shown here, and a 300 nucleotide long RNA which is known as 7S lRNA. This is a small RNA, but among a small RNA this is the largest size. That is why it is called slRNA. This acts as a scaffold, allowing the binding of the other protein subunit. SRP recognizes the signal peptide in a protein for translocation through interaction with its SRP54 subunit, which has GTPase activity as well. This SRP also binds with the help of the same subunit SRP54 with the SRP receptor on the membrane. SRP binding with SP pauses translation briefly with the help of the SRP9 and SRP14 subunits. So let us discuss SRP receptor, which is a protein present on the rough endoplasmic reticular membrane. It is made up of an alpha subunit and beta subunit. Alpha subunit is present toward the cytoplasmic side, whereas beta subunit is anchored into the membrane. It is transmembrane protein. Each subunit also binds with GTP molecules, so there are total 2 molecules of GTP bound to the receptor. So let us discuss translocon or the channel of rough endoplasmic reticulum. This is made up of 3 subunits: SEC61 alpha, beta and gamma. Alpha subunit forms the main or core of the hydrophilic channel consisting of 10 transmembrane domains. Alpha and beta, on the other hand, stabilize this channel. All the protein which are being synthesized, they co-translationally migrate into the lumen through this hydrophilic channel, and they are modified in the lumen thereafter. On the other hand, there is also a lateral hydrophobic channel which allows the migration of hydrophobic transmembrane protein to migrate directly into the endoplasmic reticular membrane. In addition, it has also several other associated proteins such as TRAM, TRAP, and signal cleavage complex or signal peptidase enzyme is there. So now we know each component, its structure and function. Therefore, let us discuss now mechanism. So this is ER membrane. Embedded into it is a translocon or the channel through which protein has to migrate into the ER lumen, and this is SRP receptor. This is ER lumen. This is cytoplasm. Protein synthesis start in the cytosol on free ribosome, and because signal peptide is present in the N-terminal region, therefore this sequence will come out first. Therefore, those ribosome which are involved in the or engaged in the synthesis of SP carrying protein, they will be migrated, they will be attached with the translocon. How it happens? When the signal peptide comes out, it is captured by a protein RNA complex known as SRP or signal recognition particle. It holds the translation briefly and allow it to diffuse. The whole complex will diffuse and docks on the SRP receptor, which undergoes conformational change, which is translated into the SEX61 component, which form the channel and allow the binding of or the placement of ribosome on the channel through which the protein translocate. Now when the ribosome has been placed on the channel, it assumes its normal rate of translation. SRP leave the site. It requires 3 GTP molecule, and so it is energy dependent. So protein will migrate through the hydrophilic channel into the ER lumen. In some cases it is often, or in some cases it is cleaved by signal peptidase. Therefore, in mature protein you will not see signal peptide sequence. However, this is not going to affect the function or its target. It will behave normally. However, if there is a problem in the SP is mutated at this stage before binding to SRP or to the channel, it will cause problem. It will mistarget the protein and causes diseases. Now the protein which are hydrophobic, they like transmembrane protein, they migrate through a lateral hydrophobic channel in the translocon, and such proteins are integral membrane protein. They migrate laterally into the rough endoplasmic reticular membrane, while the hydrophilic proteins, they pass through the hydrophilic channel, and they end up in the lumen where they are modified. So lysosomal protein, secretory protein, membrane protein, not membrane, the Golgi and ER protein or proxysomal protein, they end up in the ER lumen where they are modified according to their sequence, and then they are targeted to their site. So this is all about mechanism of translocation into ER. You might be interested to know why the protein has to migrate through the translocon into and enter into the ER membrane, ER lumen. This is because these proteins which I listed in the beginning, they are all modified, and the modifying enzymes are present in the ER. These proteins are modified according to their sequence and subsequently in the Golgi apparatus, from where they are sent to their respective site. So what type of modifications happens in rough endoplasmic reticulum? The first one is annealing glycosylation. This happens in case of secretory protein and the membrane protein. Polymerization or the assembly of some protein, they have more than one polypeptide. They are assembled in the rough endoplasmic reticulum also. Protein folding also happens. In fact, ER is loaded with a high percentage of folders or folding protein. They are called chaperones. They assist in folding and they help the protein to acquire proper folded structure. There is quality control also there. Only the protein which have folded properly and they are suitable, they are allowed to exit the ER. The remaining one which are faulty, they are retained and they are degraded in proteasome later on. So in summary, the ribosome which are engaged in the synthesis of proteins carrying signal peptide, they are connected to the translocon present on the rough endoplasmic reticulum. Proteins are modified and subsequently they are sent to Golgi apparatus. They are modified there and from there they are sent to their respective site, to ER, to Golgi, to membrane, to secretion, to lysosome and to peroxisome. So all these proteins, peroxisomal, lysosomal, secretory, membrane, ER, Golgi apparatus, they have to pass through this translocon, and that is what we have discussed in detail. And I hope that you understand the whole concept of translocation of protein through the ER channel. So thank you very much for watching the video. I ask you to like it, share it and subscribe it. Until next time, you take care. Bye bye.